Some scientific mysteries are kind of like a locked room puzzle, right? We know who the culprit is, we just can't figure out how they did it. For nearly 30 years, that's been the exact story with Huntington's disease. We've had the genetic smoking gun for ages, but the case for a cure? It's just remained stubbornly cold. But some recent breakthroughs have finally given investigators a whole new set of keys. Today, we're going to see how they're finally cracking this case wide open.
So, first things first, let's take a look at the case file. Huntington's disease. It's a devastating inherited disorder. It's progressive, it's currently incurable, and its main target is the brain. We know the cause. It's a specific genetic mutation, basically an error in a gene called HTT. This faulty gene creates a mutant protein that builds up in brain cells, and over time it causes them to malfunction and eventually die. The investigation really kicked off with a massive breakthrough way back in 1993. That's when the genetic cause was pinpointed. Then, by the 2000s, studies in animal models seemed to point to a really promising strategy: boosting the cell's own natural cleanup system. It really felt like a solution was just around the corner. And yet, here we are, decades later. The case has gone cold, and the central mystery of how this all goes down inside actual human cells remains a puzzle.
And that, right there, brings us to the biggest question we're tackling today. If we identified the genetic perpetrator so long ago, why has it been so incredibly hard to find an effective treatment for patients? Well, as it turns out, the answer wasn't in the clues themselves, but in the witnesses we were interviewing.
Okay, this is where the story takes a really fascinating turn. To get a fresh look at this cold case, scientists realized they had to stop relying on the old evidence. They needed to build a completely new way to watch the crime as it was happening. They needed a much better brain model.
You see, for years and years, the main witnesses in this case were animal models, like mice. But here was the problem. They were pretty unreliable. As the source material puts it, they were poorly replicating the human disorder. I mean, you think about it. These models often used young animals with really artificial versions of the mutation. That's a world away from a typical human patient. The investigation was basically being led astray by some really distorted testimony.
So scientists developed this absolutely revolutionary new technique. They started by taking simple skin cells from adult Huntington's patients and also from healthy individuals. Then they used this powerful cocktail of proteins called transcription factors. You can think of them as biological master switches, and they used these to directly reprogram the skin cells, basically flipping their identity, poof, straight into that of a neuron. And here's the truly brilliant part. These new induced neurons weren't just generic brain cells. They carried the patient's exact genetic makeup. And, get this, they even retained the biological age of the person they came from.
And this difference, this is the key to the entire investigation. On one side, you've got the old animal models, young, artificial, just not quite right. And on the other, you have this new human model. It's age-appropriate and genetically precise. So for the very first time, researchers had a truly reliable witness to the cellular events of Huntington's.
All right, so with this powerful new model in hand, investigators could finally get a clear, unobstructed view of the cellular crime scene. And what they found wasn't just some simple breakdown. Oh no, it was a complete and total traffic jam.
So the first clues started rolling in from a really deep analysis of all the proteins inside these new neurons. And right away, the data flagged two huge problems. First, they saw alterations in something called the AMPK pathway. Think of this as the cell's energy sensor. The foreman who realizes supplies are low and tells the cleanup crew it's time to get to work recycling old parts. At the very same time, another key player, a protein called BECN1, kind of like the manager who signs off on the cleanup job, was turned way down. Both of these clues pointed directly to a major problem in the cell's waste disposal system.
And that system has a name. It's called autophagy. And it is absolutely essential for a healthy cell. It's the process where a cell finds old, damaged, or toxic parts, wraps them up in what's basically a biological garbage bag, and then breaks them down for recycling. It is the ultimate cellular cleanup crew.
Now, as compelling as all that protein data was, the real smoking gun wasn't hidden in some spreadsheet. It was something the researchers could see with their own eyes, right down the microscope. The evidence was hiding in plain sight.
And this quote from the study just perfectly illustrates what they saw. It describes a subcellular neurite-specific impairment. So let's break that down. Neurite-specific means the problem was happening in the long branching arms of the neurons. And the problem itself, an accumulation of autophagosomes. Those are the biological garbage bags we were just talking about. They were just piling up, completely stuck in the neuron's arms, unable to get to the cellular recycling center. It was like a massive garbage strike, happening right where it could do the most damage.
Okay, so the evidence is crystal clear. There's a huge pileup of cellular trash. The obvious solution would be to just ramp up the cleanup process, right? Well, this is where the whole investigation took a completely unexpected and pretty counterintuitive turn. I mean, this is the logical next question, isn't it? For years, the main theory was that if we could just supercharge that autophagy process, send in more garbage trucks, we could clear out the toxic protein and help the cells. It was the whole basis for so many promising studies in those old animal models.
So to test this, the researchers used a drug called rapamycin, which is a really well-known autophagy booster. The hope was simple. They would come in and clear that logjam. But the reality was a complete and utter shock. In these accurate human neuron models, the treatment didn't just fail to help, it actually made the problem worse. It increased the pileup of trapped waste in the neurites. The very strategy that looked so good in animal models was completely backfiring in a human context.
This led to a huge realization. The problem wasn't a shortage of garbage trucks. The cell was making plenty of those autophagosomes. The issue wasn't a lack of effort. It was a total logistical breakdown. A massive traffic jam was preventing those garbage trucks from ever reaching the cell's recycling plant, which is way back in the main cell body. Boosting autophagy was like sending more and more trucks onto a highway that was already at a dead standstill.
So this changes everything. You see, the focus can't just be on turning on the cleanup process. The real puzzle to solve is figuring out what's causing this specific neurite-choking traffic jam in the first place. And the answer, it turns out, is a devastating 1-2 punch. First, you've got a gain-of-function problem. That toxic mutant Huntington protein is physically there, and it's actively gumming up the works, blocking the transport pathways. But at the same time, there's a loss-of-function problem. The cell is also missing the normal, healthy version of the HTT protein, which, it turns out, plays a key role in directing this very transport process. So the bad guy is blocking the road, and the traffic cop is nowhere to be seen. And this microscopic gridlock, it isn't just some abstract concept. It has a real, measurable, and seriously damaging effect on the neuron's physical structure, on its ability to communicate. You can actually see the damage it's causing. And the consequences are just plain to see. If you look at this chart, it shows the complexity of the neuron's branching structure. See how healthy neurons are rich and complex? Well, the neurons from Huntington's patients are significantly stunted, much less complex. And here's the crucial part. When scientists deliberately block autophagy in healthy neurons, they see the exact same thing happen. It's a clear link. The traffic jam directly causes physical damage, a feature that tragically mirrors what's actually seen in the brains of patients.
So after 30 long years, this cold case is finally heating up, and it's pointing investigators in a completely new direction. Maybe the key isn't about sending more garbage trucks into an already impossible jam. Maybe it's about finding a cellular tow truck, a therapy designed specifically to clear the roads, to finally allow the cell's own cleanup crew to get back to doing its job.